Good morning, students. Today we are going to discuss role of RNAi machinery in heterochromatin formation and gene silencing. I have already discussed gene silencing and there are different mechanisms which are involved in this particular process. We have discussed siRNA, we have discussed miRNA. So let us see how this heterochromatin formation also influence this gene silencing and what is the function of RNAi and this heterochromatin formation. Now, you must be knowing heterochromatin is a tightly packed form of DNA or condensed DNA which comes in multiple varieties. These varieties lie on a continuum between the two extremes of constitutive heterochromatin and facultative heterochromatin. So we are going to discuss how the role of this heterochromatin, gene expression and how this RNAi mechanism is related with this heterochromatin and gene silencing. In 2002, evidence began accumulating that implicated that RNAi machinery in heterochromatin formation and gene silencing. That means we knew that heterochromatin formation is, that means a condensed and the tightly packed chromatin. That area is not easily accessible for RNA polymerase. So the transcription efficiency is less, that we knew. But what you didn't know that is there any role of these RNAi and the heterochromatin formation? That evidence started coming in the year of 2002. This is known as transcriptional gene silencing as well as RNAi itself. Then investigator found that siRNA induced gene silencing that target a gene's control region through DNA and histone methylation. So we also know that histone methylation is another process of enhancing heterochromatin formation. So the role of siRNA induced gene silencing and bringing those enzymes which are inducing histone methylation may be a role of RNA interference as well as heterochromatin formation. Is RNAi responsible for histone methylation and resulting heterochromatinization of the centromere? That was the question. Various experimental data suggested that RNAi is involved in heterochromatin silencing at the centromere. So we will focus centromere and that how this silencing is happening with the help of RNAi we are going to study. Now Martinez and his colleagues proposed that the abundant reverse transcript of OTR region base pair with the forward transcript produced occasionally by RNA polymerase or perhaps RNA dependent RNA polymerase to form trigger double-stranded RNA. Let us explain this thing. So we are now concentrating the OTR region of centromere. This is a particular sequence in the centromere where some reverse transcript, that means imagine two DNA strands. From one strand transcription is happening. If the both strand transcription is happening, so RNA which will be formed, they are complementary to each other. Remember, So in one is the reverse transcript, another one is the normal, which for the forward direction. So if two type of transcripts are present in a particular region, so what will happen? As they are complementary, they are now forming a double-stranded RNA. Now, if there is a double-stranded RNA, that may be a target of dicer. You remember we have discussed dicer when we studied siRNA. Dicer then digests this double-stranded RNA to produce small RNA that we call siRNA. And the siRNA then associate with the protein. You know that slicer protein, Argonauts protein, AGO1 in a complex which we call RNA-induced transcriptional silencing complex or RITS. Again I repeat, we have started discussing how this heterochromatin formation and RNAi, what is the relation between these two. So we started with a particular region. Which region? OTR region of the centromere, where surprisingly we found both strands of RNA, they are forming a double-stranded RNA, and that's the target of dicer. Now, this RNA-induced transcriptional silencing complex then attacked RNA-dependent RNA polymerase in a complex known as RNA-directed RNA polymerase complex, which amplifies double-stranded siRNA. If you remember, I discussed the amplification of siRNA. Exactly the same mechanism here is happening. Dicer cut the double-stranded RNA in a small RNA that we call siRNA. This small piece will go and bind, and with the help of polymerase, it will again make a full-length double-strand RNA that will be again target of this dicer and produce many number of siRNA. This second point is the amplification of siRNA. Then RITS, RNA-induced transcriptional silencing complex, then causes recruitment of histone 3 lysine in 9 methyl transferase. So that's the evidence that this complex only bring this enzyme which will methylate the histones at the lysine amino acid residue. Methylated lysine 9 recruits SWI6. So I believe by this time nucleosome structures and remodeling already been taught. So you must be knowing this protein name SWI6. This is a protein which is helping heterochromatin formation and chromosome condensing. So once this enzyme, this particular protein is recruited, which is required forming heterochromatin. Carefully visualize this diagram, what we are talking here. We are discussing this OTR sequence of centromere where few these SWI6 proteins are bounded. It's not enough to form a heterochromatin. So what is happening? Reverse RNA are already synthesized. This is double-stranded RNA center. This is DNA where this SWI6 are bound. That is the DNA. So you can see in one line the forward transcript is present, which is in a dotted line. In the other side, reverse RNA is showing there. So this both the strand of DNA can act as a template. That's why you are getting this double-stranded. The moment you are getting a double-stranded RNA, this is the target of dicer. You must be knowing this thing because I have discussed this thing. So with the help of AGO1 argonates, it will form siRNA. So what you are seeing here, RITS. Now this step E, number E step is an amplification step. This small will go and bind again any RNA, and that with the help of RNA dependent RNA polymerase, it will extend and give another double strand of RNA that will be target of dicer. Again it will cut, again it will form Si. So this is an amplification step. Once you get, this is one your complex guide RNA which we call with the protein, and these are the plus N. That means so many numbers of you have the amplification happen. So that will recruit whom? Histone transferase H3 methyl transferase. So this is recruited here, HMT, histone methyl transferase. It will recruit. The moment it will recruit histone methyl transferase, it will recruit many more SWI6 protein. Binding of this protein with the help of, in presence of HMT, that means histone methyl transferase, will trigger the heterochromatin formation. The exact thing I have discussed in the next slide. The outermost region, that is OTR, of the centromere is constantly being transcribed to produce reverse transcripts, and production of forward transcript probably also occurs at a low level. Okay, so we are getting both the sides RNA from both the strand of DNA. After transcription and reverse transcription, or after reverse transcription and RNA dependent RNA polymerase, that is the amplification, we have double-stranded RNA. So we are getting double-stranded RNA. Dicer then cut this double-strand RNA and form siRNA. You know that with the help of AGO protein, which I'm describing this diagram here. Yellow colored protein means the AGO proteins associate with the single-stranded siRNA to form an RITS. Then this RNA dependent RNA polymerase and this full complex amplifies the siRNA, producing more number of double stranded Si. The moment we get the amplification, the silencing will be more. The RITS through its siRNA associate with the OTR region, either through direct interaction the DNA or through interaction with the transcript in this region. Then RITase, this is the RNA induced transcription silencing complex, attracts an histone methyl transferase to OTR. Histone methyl transferase, that is the lysine 9th position of get methylated. Of course, the histone is part of nucleosome, which is not shown here for simplicity. Then what happened? The methylation in turn attracts that SWI6 protein, which is shown in red color. If you see here, these are the protein, and then this helps to spread heterochromatinization. So we have shown here that how siRNA induce heterochromatin formation in certain region. Now, another experiment we are now going to study, which is silencing by siRNA targeting EF1A control region. So far, if you have gone through siRNA, you have studied, we have started telling that you have to design your double-stranded RNA from the coding sequence, not the promoter sequence. If you want to design siRNA, remember that was the first criteria. But there's something we are going to now study. The exception is also there. This is EF1A. This is an eukaryotic translation factor. That control region, we have taken control region meaning promoter sequence and the upstream sequence of this particular gene. Which gene? EF1A gene's control region they have taken, and they have cloned a known reporter gene, that is GFP, green fluorescence protein. Listen carefully. They have prepared a construct. Which construct? They have taken the promoter sequence from EF1A. This is the control region. Under this promoter, they have cloned a reporter gene. Which reporter gene? The reporter gene is GFP. GFP meaning green fluorescence protein, which you can measure its transcription. Okay. Now this they have constructed inside a virus vector. So that cell is called FIV vector. Why we have constructed a virus base? Because its efficiency of infection is more. This process we will call transduction. So efficiency is very, very high. It will go to nucleus and it will integrate. This gene will integrate inside the cell line they have taken, the human cells they have taken, so that will be integrated in the human genome. Okay, so one more thing, this virus infection will make the nucleus pore little bit more efficient to allow other small RNAs. Now again I repeat, this is a construct, this is a construct they have prepared. What is the construct? That they have taken a control sequence meaning the promoter sequence, okay, from EF1A gene, and they have cloned a reporter gene, green fluorescence protein, and the whole thing is in a viral vector. Okay, they have transduce, we should say, this vector in the human cell line, and hopefully it will go inside the nucleus and it will integrate in the genome. After that, they are doing a real-time PCR in presence of one SI, two types of SI. One SI they have designed from reporter gene, that means from the GFP region. Another SI they have designed from a control sequence. Which is the control sequence? This EF1A control sequence. Now let us read it, then I will again explain. So what they are showing here, GFP mRNA expression, real-time PCR, that means how many transcript been produced that you can quantize, and that they have showing. So two panels are there. One is in presence of the drug, one is in absence of the drug. Which is the drug? TSA is a drug. TSA, trichostatin A, that is a drug which will inhibit histone function. Another drug also 5-AZA-C, that is also inhibit the histone's function. So what is our objective? Objective is to understand silencing by siRNA targeting EF1A control region, whether this SI which been targeted from this control region is inducing heterochromatin formation and silencing the gene. That's why they have taken two types of SI, one from the sequence of GFP, that's a normal SI, understood? And when I'm designing an SI from the control sequence that I'm giving, so that will give, if the silencing takes place, that we can mention that the control region also can, the heterochromatin formation takes place, and that's the reason the silencing happening. So let us see silencing by an SI targeting EF1A gene control region. So there are two type of SI they have designed. One is EF52. EF52 is an SI RNA name which they have designed against the control region, that means the promoter sequence. Which promoter sequence? EF1A gene. And another one they have designed from the reporter gene, that is GFP. Okay, so real-time PCR assay of GFP mRNA in human cells bearing a GFP gene driven by EF1A gene promoter enhanced region. Understood? Cells were transduced with FIV bearing, FIV is a virus bearing the GFP gene construct, and then siRNAs were added in the absence or presence of drug. Which is the drug? TSA and 5-azacine. Then real-time PCR was performed to measure the concentration of GFP mRNA. You can quantize. The bars shows the result with no SI control and SI that targets the coding region of mRNA, that means GFP, and the SI that targets that control region, EF1A gene control region. Let us see the first panel. We will discuss the first panel. No drug has given. So the normal thing what we are seeing. Control meaning no, we have not given any SI. So what we are doing, we are checking the how much GFP that mRNA transcription takes place. Okay, it is already integrated hopefully inside the genome, so we are doing an only real-time PCR with the primer. So we know that how much that particular specific GFP mRNA expression we are checking. So in the control we are seeing a good amount. The first one value is given. Now the second one, here we have treated the cells with the SI, which it is targeted which sequence? GFP sequence. You see the decrease, how much it has decreased. The third one is again we have treated with another SI, where which SI? This SI is designed against the control region, meaning the promoter sequence of EF1A, under whom this GFP is expressed. Okay, you see there is no expression at all. Now the same experiment they have done, that is the blue color panel, in the presence of TSA. So question was, this silencing what is happening in the control region SI, is it because of heterochromatin formation? So if the heterochromatin formation is involved, then the drug which is I am stopping the histones, okay, that will definitely have an effect. So the drug is TSA, which will not allow histones protein to synthesize. So what we are doing here, in presence of that, they have done the real-time PCR. Control, there is no more effect. There is again silencing in the GFP, but when we are GFP, that is this SI treatment in the coding sequence, but this EF52 is the SI which is designed against whom? Against this control sequence. What we are seeing, it has not been silenced. So the moment we treat the histones, antihistones, some drug, you are seeing that is not, the transcription level is so high. That means in the previous panel, that means the red colored panel, you see this SI was working through the heterochromatin formation. The moment I stop that effect with the treatment of TSA, there is no effect of SI at all. So hope you understood. And this experiment they are showing that under the same condition, they are measuring the RNA level. That is a simple northern blot they have done here. This is the control gap dehydrogenase. So that is a control. So for both the SI treatment and the control, they are seeing the other mRNA, which is a housekeeping gene, is steadily forms, but when we are seeing the GFP in presence of this siRNA, we are seeing it is going down. So silencing happening, then that too happening in the RNA level. Okay, that this experiment proves that in the RNA level only silencing is happening, but the previous experiment is showing that yes, RNA level definitely, because we are checking here real-time PCR, but see at least which one affecting the control region, there is a heterochromatin formation. So silencing by siRNA targeting this EF1A control region, this silencing was happening through then heterochromatin formation, because in presence of histone, the drug which is affecting the histones, this silencing effect is completely abolished. So hope this much is enough for you. Thank you very much for listening.